This animation provides a general representation of an animal cell as it progresses through interphase, mitosis, and cytokinesis. In interphase, the chromatin in the nucleus is highly dispersed and individual chromosomes cannot be distinguished. Each chromosome replicates to produce two identical sister chromatids, which remain attached to each other. Although the centromeres also replicate at this time, they do not separate until later in mitosis. The centrioles, which organize the interphase array of cytoskeletal microtubules, also replicate during interphase. Transition from interphase to prophase occurs almost imperceptibly as the chromatin begins to condense into discrete chromosomes. The diploid number of chromosomes in this cell is 6, consisting of 3 pairs of homologs colored red, blue, and purple in this illustration. Near the end of prophase, the cytoskeleton breaks down and the mitotic spindle begins to form between the duplicated centrioles as they move apart to opposite poles. In the transition to metaphase, sometimes referred to as prometaphase, the nuclear membrane breaks down into small vesicles, which disperse throughout the cell. Spindle microtubules from the 2 spindle poles enter the nuclear region and attach to the chromosomes at the centromeres. Each chromosome can be clearly seen to consist of 2 sister chromatids joined at the centromere. Eventually, spindle fibers coming from the 2 poles attach to the sister chromatids of each chromosome and pull them in opposite directions. Metaphase begins when this tension aligns the chromosomes at a single plane, called the metaphase plate, midway between the poles. The chromosomes remain bound together along the entire length of their sister chromatids. Anaphase begins suddenly, as the centromeres split and the sister chromatids separate, each being pulled toward the spindle pole it faces. As the chromosomes approach the poles, the spindle fibers between the poles elongate and the 2 poles move away from each other. During telophase, chromosome movement comes to an end. The spindle apparatus disassembles, and the nuclear membrane reforms around the chromosomes. Within the nucleus, the chromosomes decondense. Mitosis, the process of nuclear division, is now complete. Each daughter nucleus has received exactly 1 copy of each chromosome. Upon completion of cytokinesis, 2 separate daughter cells are produced.